Good morning, everybody. Welcome to what will be the last session in the BMGF COVID webinars for a while, as we'll take a break over summer. But I'm very pleased to welcome Martine Keenan from Epichem to be our final speaker of the session. Martine hails from the UK, but is now in Perth in Australia, where she's been for quite a long time now. She did her degree and PhD at King's in London, and then a postdoc in Germany. She worked at Eli Lilly before moving to Australia, and has been working as part of Epichem on a wide range of global health projects. And today she's going to tell you about the role of CYP51 inhibitors in Chagas disease and learnings from that area. I'll hand over to you, Martine. The floor is yours.
Okay, thank you very much, Caroline. Good to be with you all, and thanks for the opportunity to share some key learnings from the discovery programs that we've worked on in the Chagas field over the years. So first of all, I'd like to start by introducing you all to the team. These are the guys that have done the work that I'll share with you today. So the work is the output from a drug discovery consortium that was established by the Drugs for Neglected Diseases Initiative more than 10 years ago. And you may know that DNDi is a non-profit research and development organisation that is developing new treatments for neglected diseases. And the purpose of this consortium was to develop new treatments for Chagas disease. So the core members are captured here, and we represent the multidisciplinary effort of a drug discovery program. So Epichem does the chemistry and medicinal chemistry. We have in vitro biology at Griffith and Murdoch. The pharmacology is done at CDCO in Melbourne, and then in vivo biology done at London School of Tropical Medicine. And other partners and collaborators DNDi have in their global network. So what is the role of CYP51 inhibitors in Chagas disease research? So I'm actually going to start with the 4 key points that I hope that you'll take home from this presentation. So first of all, research focused on the development of cruzi CYP51 inhibitors, I believe, has been instrumental in progressing the Chagas disease research field. As recent as 5 years ago, cruzi CYP51 inhibitors led the pack as potential new drugs for treating Chagas disease. And their accessibility and expansive use in basic research has facilitated new understandings of parasite biology, parasite host interplay, and importantly, assay development to facilitate fast-paced pharma-style R&D programs able to accelerate discovery efforts in this field. Secondly, it's important to note that compounds inhibiting this enzyme have actually failed to demonstrate curative activity for Chagas disease in the clinic. And so despite the significant antiparasitic activity shown by a wide variety of CYP51 inhibitors in vitro and in vivo, the leading Azole antifungal inhibitors that were repurposed a few years ago now, posaconazole and replication, showed limited curative potential in randomized controlled trials. So hopefully we'll uncover why this is in this presentation. And thirdly, the cruzi CYP51 is a promiscuous enzyme accommodating a wide variety of structural fragments used by medicinal chemists. And I think it's important for us to remember this. It's one possible reason for why CYP51 inhibitors have been at the forefront of research for so long. It could be the fact that they're very easy to discover. Many CYP51 inhibitors are inadvertently selected as hits of interest from phenotypic screens because these are mechanism agnostic, and many common drug-like fragments added during the course of a compound optimization program predispose compounds to operate via this mechanism. And lastly, a common response fingerprint is discernible and useful to keep in mind when evaluating the output from phenotypic screens. So on the point of knowing what you are working with, when you screen blind with respect to mode of action, I think it's possible to work out what compounds are working by CYP51 inhibition and take steps to eliminate these molecules from your program, or proceed with the knowledge that this is the likely mode of action. Okay, just briefly on the disease itself. What is Chagas disease? Well, Chagas disease is a potentially life-threatening illness caused by a protozoan parasite, Trypanosoma cruzi. It's found mainly in endemic areas of continental Latin American countries and is a major public health problem. Over the last 20 years, globalization has spread the disease to other continents, such as the United States, Canada, many European countries, and some African, Eastern Mediterranean, and Western Pacific countries. About 6 to 7 million people are affected worldwide, with approximately 12,000 deaths attributed to Chagas disease per year. In Latin America, the insect vector is a beetle, and from the family Triatomoni, commonly known as a kissing bug. And transmission occurs to humans via the urine or feces of the beetle, which contains the parasites. And this happens when it takes a blood meal. So after biting an exposed area of skin, usually the face, the beetle defecates or urinates close to the bite wound. And then the parasites are smeared into the bite, the eyes or the mouth, or into any skin break and enter the circulation. So other routes of transmission are oral, so contaminated foods, in blood transfusion, congenital, and sometimes in organ transplantation. The life cycle of Trypanosoma cruzi is shown here. So on the left-hand side is the insect stage, and on the right-hand side is the human stage. So if we just focused here, once the parasites enter the bloodstream after the beetle's taken a blood meal, metacyclic trypomastogotes, which is one life phase of the parasite, are released near the bite wound and invade cells close by. Once these trypomastogotes are inside the cell, they differentiate into amastogotes, which is a round form of the parasite. And then over a period of 2 to 4 days, the amastogotes multiply by binary fission and differentiate back into trypomastogotes, are released back into circulation when the cells burst, and the infective cycle continues, leading to the clinical manifestations of Chagas disease. So a couple of important points to remember. T. cruzi is an intracellular parasite, a key point of difference to African trypanosomes, which cause sleeping sickness. Trypomastogotes can infect almost any nucleated mammalian cell, and this is a key problem for the development of therapies. Only the amastogotes can replicate, and this life stage is generally the main target of interest for drugs and in drug screening programs. And research continues to reach new understandings about the various life stages of the T. cruzi parasite, not just simply trypomastogote or amastogote. And this is in order to describe some of the nuances of the infection, such as its persistent nature. And a dormant variant of the amastogote has been proposed to explain this behaviour. So if we look on the right-hand side of the slide, the Chagas disease presents itself in 2 phases, the acute and the chronic phase. The acute phase lasts for about 2 months of infection, and parasitemia is very high. So the number of parasites that circulate in blood slowly decrease over time, and symptoms are absent or mild or unspecific. So in less than 50% of people bitten, the first visible sign will be a skin lesion or a swelling of the eyelid, and occasionally fever, headache, and flu-like symptoms. But during the chronic phase, the level of circulating parasites is low or undetectable, and the parasites are kept in check by the immune system, and they remain hidden in tissues. And Chagas disease sufferers can remain infected, but asymptomatic for life. Interestingly, about 30% of patients then go on to develop cardiac disorders, and up to 10% suffer from digestive disorders, typically enlargement of the heart or the esophagus, and sometimes neurological alterations or mixtures of conditions. In later years, the infection can lead to sudden death due to cardiac arrhythmias or progressive heart failure caused by the destruction of the heart muscle and its nervous system. So really, in summary, in 2005, the World Health Organization recognized Chagas disease as a neglected tropical disease, and this facilitated greater recognition of the disease on the international scene and improved commitment to a range of factors, including R&D, treatment, prevention, and education. There are some treatment options available currently, but these are limited to 2 compounds, benznidazole and nifurtimox. The compounds were introduced in the 60s. They're given orally, and they have significant toxicity associated with their use. Benznidazole is the first-line treatment, as the side effects of this drug are less severe than with nifurtimox. Benznidazole is active in the acute phase of Chagas disease with a cure rate of up to 80%. Curative capabilities in the chronic phase are limited, but use is advocated as some studies indicate treatment can reduce cardiac changes and delay worsening of the clinical condition of the patient. So better to be taking the drug than not. It's also been proven to be effective in reactivated T. cruzi infections caused by immunosuppression or immunosuppressive therapy related to organ transplants. So the compounds work by enzymatic activation of type 1 nitroreductase, an enzyme found in some protozoan parasites but not in humans. It generates free radicals and inhibits DNA synthesis in both mastigates and tripomastigates. The generation of reactive metabolites on exposure to benznidazole is responsible for a well-documented spectrum of idiosyncratic adverse drug reactions, which can lead to treatment interruption or discontinuation by a significant percentage of the patients. As much as 30% of patients will stop taking the drug. And the symptoms are particularly problematic when the compound's given in high dose. On the flip side, benznidazole is a cheap drug to manufacture, so 2 steps, no chiral centers. It's not that active against T. cruzi, so micromolar in vitro activity, but it does have a long half-life. The dosing regime is intense, so 5 to 7.5 mg per kg per day, but 2 to 3 times a day for 60 days. So patient compliance can be an issue when taking this drug. And as I noted, there are a number of adverse reactions: headache, anorexia, weakness, skin rash, and GI problems in particular. The side effects are commonly described in the first few weeks of treatment, but they decrease as treatment progresses and are generally reversible upon drug withdrawal. So the activity of the trypanosome nitroreductase is very low in children and young adults, and this explains the fewer adverse side reactions in these age groups. And benznidazole has just been made available in pediatric formulation for children. Just on the right there, a couple of words on nifurtimox. It's rarely prescribed. It's a backup option because its side effect is more pronounced. So with these 2 drugs alone, it's obvious there's a clear need for more therapeutic options for Chagas disease. And this is where the Azole antifungal drugs come in and repurposing opportunities. So drug repurposing is an excellent strategy to pivot new treatment options into the clinic, and this can occur much more quickly. And the considerable impetus for progressing these azole antifungal drugs as treatments for Chagas disease came from the group of Julio Urbina in the late 1990s. And he was looking at a triazole antifungal D0870 that demonstrated really superior results over nifurtimox and ketoconazole in acute and chronic in vivo models of T. cruzi infection. And that was way back in 1996. Unfortunately, the development of this drug was halted because of adverse cardiac events in the clinic. And then the quest for a suitable azole to treat Chagas disease was on and shifted to alternative compounds. And the most successful effort behind the push to advance these compounds into the clinic has involved this compound here on the left, posaconazole. It's a broad spectrum antifungal drug approved in 2006 by the FDA as a salvage therapy for fungal infections in immunocompromised patients. And the compound showed remarkable in vitro activity in amastigotes, so really low nanomolar activities in vitro. And 30 to 100-fold more potent than other well-known azoles like itraconazole and ketoconazole. It also provided very high levels of parasitological cure in acute and chronic mouse models of Chagas disease at the time. And so to support advancement of this compound at the time, there were many head-to-head studies against benznidazole, as they were profiled against a range of T. cruzi strains in both acute and chronic in vivo infection models, and posaconazole outperformed benznidazole. So why was this? Well, the theory at the time was it was a combination of the high intrinsic activity and really excellent pharmacokinetic profile. So posaconazole has a very long terminal half-life and a high volume of distribution. And it was this view that it would enable drug penetration into multiple infected tissues. Another compound of interest at the time, at the bottom there, ravuconazole. In fact, E1224, a monolysine prodrug of ravuconazole, was being progressed by the DNDi and Assay, and entered phase 2 trials in 2011. Again, the in vitro activity of ravuconazole is very impressive, picomolar activity against intracellular amastigotes. And although the PK was not so great in mouse models, the compound has had a very long half-life in humans, on the scale of 4 to 8 days, and that was sufficient to convince the teams that this needed to be progressed into the clinic. So azole antifungal drugs work via the inhibition of sterol metabolism, and sterols are important constituents of cellular membranes and are essential to normal structure and function. So cholesterol is the main sterol found in mammalian membranes, but ergosterol and other sterols predominate in eukaryotic microorganisms such as fungi and protozoa. So CYP51, or sterol 14-alpha demethylase, is a hemoprotein, and it catalyzes the 3-step oxidative removal of the 14-alpha methyl group of cyclized sterol precursors, leading to the production of cholesterol in humans and ergosterol in fungi. And the commonality of the sterol biosynthetic pathway between fungi and protozoa facilitated the repurposing strategy for approval of approved azole antifungal drugs and compounds of clinical interest to be investigated for the antiparasitic activity. So when we started to look at this area, we were aware of one major drawback of using the azole antifungal drugs, and that was their potent activity against CYP3A4, which is a major drug metabolizing enzyme, and hence their potential to cause drug-drug interactions in patients. So to overcome this drawback, we were interested in investigating other fungal CYP51 inhibitors that didn't contain an azole, and we looked at herbicides and insecticides as a source of new compounds. And after a few targeted screens, we identified a compound called phenaramol, which is an antifungal herbicide used for controlling powdery mildew in fruits and vegetables. And although we knew its antifungal mode of action was via CYP51 inhibition in fungi, it was an inference that this was a mode of action against T. cruzi, and something we're able to investigate and determine later on in development. So some other compounds of interest at the time, when we started our Hit to Lead campaign, the tipifarnib series from Mike Gelb and Fred Buchner, which is a repurposed anti-cancer agent, and the LP10 series from Larissa Podos, which I believe came from a TB effort. So there was a number of people looking at these types of molecules at the time. And just have a quick look at the crystal structure of CYP51 from T. cruzi, which was solved as we were working on this effort back in 2010. So you can see the various alpha helices have been coloured here in diagram C on the left. And if you switch to a distal view, you can actually see the heme ion in the middle there, and the iron atom. And this is the substrate access channel where ligands bind. And on the right-hand side here are actually some of our early candidates that we were able to get a crystal structure of, binding to the heme motif. And the difference between a pyridine-based CYP51 inhibitor is just the length of this bond. And it's a bit weaker than in the azole antifungals, but because there was a number of interactions between the ligands and the other elements of the protein in the substrate axis channel, we were able to make very potent compounds, and I'll show you those a little bit later on. Okay, so As you all surely know, a discovery program needs to be driven by an assay cascade, which progressively triages compounds of interest through high throughput, large volume assays in vitro towards lower throughput, slower in vivo assays. And these are designed to translate efficacy in animal models into clinical efficacy in patients. That's the idea. But only when you've been successful in the clinic can you really begin to understand how well your assay cascade translates benchtop science into effective treatments. So in 2008, when we started our investigation, we built a screening cascade based on the best tech at the time. So from design and compound synthesis, our in vitro assay was a phenotypic assay, measuring parasite kill in whole cells according to the protocol developed by Fred Buchner, so a colorimetric based assay. The readout is shown on the right here. So we're looking at increasing compound concentration versus the colorimetric output, which was translated into parasite inhibition, so 0 to 100%. Your regular tier 1 ADME assays, including CYP3A4, because we were intent on not having that cross-reactivity, oral exposure, and into in vivo model. And we had to develop this model as we progressed the effort. So on the right-hand side, this was the model that we ended up with, which is a subchronic infection model in mouse. So you're looking at days of the experiment, 0 to 60, and number of parasites in blood over time. So after mice were inoculated, drug treatment continues for a period of time, 5, 10, or 20 days. And at regular time points, parasitemia is measured after daily dosing of drugs. Then a 10-day rest period, and then you're looking to see if those parasites recrudesce and come back into the bloodstream, being able to hide in tissues, and this is done after 3 cycles of immunosuppression. So a drug that decreases parasitemia here in the solid line during dosing may well experience parasite relapse, so B, and therefore not be curative. And we're looking for something that looks like here on the dotted line where there's no relapse. And at the time, after PCR investigation of organs and tissues to see if there's any parasite DNA left, that compound would be called curative. So that was the model that we had at the time. And there are a couple of red boxes here. One was a target ID, and that we addressed a bit later on in the effort. Okay, so this really is just a quick summary of our first rounds of optimization. So we took phenaromol, which had pretty good in vitro activity for a hit, 350 nanomolar. And you can see it's a very modular structure, so we're able to interrogate each of the atoms and pharmacophores quite readily to end up with our first prototype lead compound, BS177, which had improved in vitro activity and good oral PK, which I've shown here on the right. So compound concentration over time. And you can see that we get high plasma concentrations relative to the IC50 and extended exposure out beyond 24 hours. And measuring the in vivo activity using the protocol I just described, BS77 showed a good suppressive parasite action in the early phase of dosing. Parasites remained undetectable during the rest period, but as soon as the mice were challenged with cycles of immunosuppression, you can see the parasite rebound occurring, indicating that there was no cure after we'd done the PCR. So active, good mouse survival, but no cure. And so we proceeded to do a number of rounds of optimization of the molecule. And so our lead optimization effort was really focused at looking at the central portion, and we discovered that we could extend the third aromatic ring into space. So I've just shown a couple of examples here. We've got piperazine and aminopiperidine, and you can work out all the other types of scaffolds we looked at based on orientation of attachment and also ring size. So these were our leading molecules. And we were able to gain a lot of information by running through our flow scheme, looking at the activity, generating some exposure data and measuring the in vivo efficacy of these compounds. So the first useful point that we learnt was that we had a pretty flexible pharmacophore. So we could place these 3 aromatic groups in quite a large area of chemical space with a spacer in the middle, provided we had an aza atom here to interact with heme. And the second thing we learnt was we established a preliminary PK/PD assessment, so looking at the pharmacokinetic profile versus the pharmacodynamic readout. And so if you look at the graph on the left here, this is a graph of oral exposure of a number of compounds, all plotted on the same graph, so compound concentration over time. And the line indicates the average in vitro IC50 for these compounds. And so you can see here that if your compound concentration stays above its in vitro IC50 for a period of 24 hours, we were getting efficacy in vivo. And in contrast, when the plasma concentration of the compounds dipped below the in vitro IC50, around about 12 hours here, these compounds were not active in vivo. So it's quite a very useful triage for us using this PK/PD relationship as we were making compounds towards the later stages of the effort. And the same idea is captured in this bar graph over here, where we're looking at the relative compound concentration to IC50, so fold difference for a variety of compounds. And so we had a good correlation that we could use, and something that's been very useful to us moving forwards. So we were able to finish off our optimization program to produce some potential early clinical candidates using this PK/PD relationship and our pharmacophore model. And we developed 2 compounds of high interest, so 967 here and 1246. And I'll just take you through a little bit of chemistry towards these compounds. They're relatively short and efficient. So for 967, just simple wood examination, put in the pyridine and the piperidine, and then adding in the para-CF3 phenol group quite straightforwardly through cross-coupling, and then another cross-coupling step at the end there to generate our pharmacophore. Sorry, that's an SAR reaction at the end. So very quick and simple. What is interesting is some analogues here in this series were super potent. So picomolar activities in the whole cell assay is really quite unusual. So this compound was obviously making really great interactions with the target to generate these types of potencies. And a balance of the PK and the potency, we selected 967 to go forwards. So our other molecule of interest, 1246, again, very straightforward synthesis, SNAR to generate this useful nitrile intermediate, hydrolysis to the acid, and then amide formation at the end. And then with the chiral center, we just did chiral chromatography to generate our compound of interest, which we established by X-ray was the S enantiomer. So pretty straightforward, and we could scale that up quite easily. Again, a couple of the analogues here on the right, not quite as potent as the 967 series, but still really low nanomolar potencies for these compounds. And once again, balancing the PK with the potency and the in vivo efficacy, 1246 was the compound that we took forwards. So using our in vivo model, we established at the time that these compounds were very exciting because of the level of efficacy in the mouse infection model, which was comparable to both the standard of care benznidazole and the gold standard at the time, posaconazole, in the same assays. So the in vivo efficacy data is shown here. The studies were run using the protocol I described earlier, including a 20-day dosing period, a rest period, and immunosuppression stages, followed by PCR analysis of organs and tissues to detect parasite DNA. And under this protocol, the PCR confirmed curative activity was demonstrated in 60% of the mice for 967 and 50% of the mice for 1246. And the benznidazole result here for comparison is a 40% cure rate in this assay. So at the time, these compounds were state-of-the-art and considered to have potential candidate level efficacy. And as such, they were moved into further profiling. And so the summary of that profiling is here, shown for 1246. So returning to the assay cascade I showed earlier, you can see that we managed to run through and successfully tick off all of the assays in the cascade, including safety and selectivity profiling. And at that time, we had the nice crystal structure early candidates bound in with this, bound into CYP51. Importantly, we managed to avoid any CYP3A4 cross-reactivity, which was one of our primary goals. And we thought that the activity and PK profile was good enough to demonstrate what was to be considered in vivo efficacy or predictive of good in vivo efficacy in the clinic at the time. And so we nominated this compound as a potential preclinical candidate. So why potential? Because at the time, posaconazole was under evaluation in a randomized clinical trial alongside benznidazole, and we were waiting for the outcome, which, as I shared at the beginning, was actually unsuccessful, with posaconazole demonstrating a suppressive rather than curative action in the clinic. So why was that? It's obviously very important to understand what had gone wrong. And actually, it was due to one of the boxes that had stayed red during our assay cascade, and that was about strain profiling. And this actually turned out to be a very critical factor in failure of posaconazole in the clinic. So although the trial of posaconazole was conducted in Spain, most patients were actually from Bolivia. And in that country, the prevalent strain of T. cruzi is T. cruzi 5, and we'd been screening against TC6. And it was subsequently shown that CYP51 inhibitors in particular are least effective against this particular strain. So the treatment failure with high dose posaconazole was up to 80%. And there was some really great work reported in this Nature Scientific Reports paper that dived into that a little bit deeper. So I'll just show their results. So 2 possible reasons for the suppressive action rather than cidal action. And they are efficacy against the clones and speed of kill for the CYP51 mode of action. So the graphs you're looking at here on the left, you've got percentage activity, and the coloured curves represent the different types of T. cruzi strains that were investigated in vitro. These are the results for benznidazole. So you can see benznidazole has a similar IC50 against most of the clones and is fully efficacious. But posaconazole tells quite a different story here, with a variety of IC50s and less than ideal percentage activity against the parasites in vitro. And actually 1246 was also evaluated in the study, and we get a similar result to posaconazole. So that's one potential reason for lack of efficacy in the clinic. And the other is that the mode of action of sterol inhibition is quite a slow one to be effective in terms of killing the parasite. So here are time kill experiments, and this time the different colours represent different concentrations of compound, and they've been evaluated over time against, again, parasites in vitro. So you can see at the highest doses of benznidazole, you get almost full inhibition between 48 and 72 hours. But for posaconazole, the inhibition is still on its way down and doesn't really get to 0 until about 144 hours. And the same profile is in existence for 1246. So there is one other possible explanation for the lack of curative activity that was subsequently proposed in 2018. And this suggested that there was a dormant form of the intracellular amastigote phase. And this phase was thought that it can resist drug pressure. So whilst not replicating, it can actually be re-triggered to morph into the infective trypomastigote form when cells burst and hence proliferate infection. And this theory or hypothesis is under investigation now, and it's important that if we can replicate this in vitro as well as we progress new efforts. So what was the impact on the Chagas field of the posaconazole failure in the clinic and subsequently voriconazole as well? Well, really the impact was to halt a lot of the research into the CYP51 inhibitors as potential therapies for Chagas disease. Here are some of the compounds of active interest at the time, but I think the research has really tailed off on these molecules, and certainly we took the decision to stop working on CYP51 inhibitors and look for a new mode of action. But unfortunately, as we'd parked our potential clinical candidate and we started on some new hit to lead efforts, we were successfully evaluating a number of new series and finding molecules that were super potent in vitro, had great PK profiles, and were doing pretty well in vivo assays. And we had run a number of optimization programs. But if you just stop to study the pharmacophores of these compounds, you'll see a basic nitrogen and a lot of commonality with respect to the shape and chemical space that's across the set. And it transpired that when we were able to test these compounds, they were all in fact cruzi CYP51 inhibitors. So what next? How to diversify away from this mechanism to de-risk the drug discovery portfolio for Chagas disease. And this is where developments in assay technology have played a critical role in moving the field forward. The first of these is an in vitro assay. So it's an image-based cruzi inhibition assay. It's very sensitive, it's a phenotypic screen, and it's based on spot analysis algorithms in amastigotes to count the parasites in vitro. And you get a rich vein of information from this assay. So you get an efficacy, so 0 to 100% parasite inhibition. You get a 14-point dose response curve where you generate your IC50, and you get an N of 2 in each assay, and you can also look at host cell activity as well, so drug just against host cell alone. So it's, I called it a 2D readout from primary in vitro assay, and the incubation time is 48 hours. And these are the types of graphs and outputs that you can get. So with A, you can see we've got a compound with a low selectivity index. So this is activity against amastigotes, and this is activity against host cell. And this is really important because if you're not aware that you've got host cell activity, you might be over-interpreting your in vitro IC50, because if a compound can kill a host cell where a parasite lives, you could just interpret it as parasite inhibition by the compound. So that's quite a subtle nuance that has to be appreciated with potentially cytotoxic compounds. And here is an example of a cytotoxic compound in which the activity against the host cell completely overlays with the activity against the amastigotes. So being able to actually look at the graphs and the raw data is really important for a medicinal chemist, so they're not over-interpreting the in vitro data when they make their design decisions. And what was really interesting is when the CYP51 inhibitors were profiled, because of this short incubation time, you can actually see they don't clear all the parasites in vitro, so you have this sub-efficacious response against the amastigotes. And this is fairly typical for CYP51 inhibitors that have been profiled. So the other assay that's made a huge difference is a bioluminescent imaging, again an imaging assay for cruzi in mice. So it's very, very sensitive. So the issue with sampling parasites from blood is that the limit of detection was not low enough, and you couldn't really see where these parasites went in the mice as they cycle around, and they move an awful lot. So this is the output of a chronic assay, and we're looking at mice that have been infected with cruzi engineered to express a bioluminescent reporter enzyme, and they are dosed with luciferin, which is the second part that enables the bioluminescence to occur. And then over a period of time, the mice are imaged, and you can see how the infection moves around the mice and flares up and decreases. So this line at the top is the control mice, and the flux from the image is related to the number of parasites, and that's how it's used to calculate how many parasites are remaining. So B is benznidazole. So you can see mice are cured under a 20-day dosing regime with 100 mg/kg of benznidazole. And C and D are actually posaconazole, 20 mg/kg per day for 20 days. And so whilst there is some suppressive parasite activity by posaconazole, you can see that the parasites have reappeared, so a suppressive, not curative action. To enhance the readout, you can do ex vivo imaging of the mice, of the organs and the carcass, and here's a diagram of what's in the dish here. So even though you might see a cured mouse, you can really dive into some detail and detect down to hundreds of parasites. So really very sensitive technique, and really we need to get this assay validated in terms of how well it translates into clinical efficacy. But a lot more information that can be gained from this experiment, but also it's raised the bar considerably in terms of what's expected of new treatments, and that is a sterile cure, because even the lowest level of parasites can reinitiate infection. Okay, so I think what we've learnt after working in this area for a while is really how to spot a CYP51 inhibitor, and I've put some thoughts down here on this slide in terms of what a fingerprint would look like for a CYP51 inhibitor. So first of all, typically an ASA heterocycle with lipophilic side chains, and I think it looks like this tripod structure where you have the basic nitrogen that needs to be unhindered, otherwise it can't interact with the heme. And this is one of the ways you can investigate if you have a CYP51 inhibitor. You can substitute next to the nitrogen. If you lose activity, the inference is that it could potentially be the mechanism. And typically lipophilic compounds that are high molecular weight that look like this are all features that are typical of CYP51 inhibitors. As I showed earlier, the in vitro assay can be really instructive in terms of whether you have a CYP51 inhibitor, so it's kind of weight of evidence with structure and the in vitro output. So it's really important to be looking at the raw data from your graphs, from your biology colleagues, if you're a medicinal chemist, and typically efficacy is less than 90% in these short-term assays where drug incubation is 48 hours. Also, CYP51 inhibitors tend not to be active against trypanomastigotes. So if you're able to do that assay as well, and the compound's not active, that's also suggestive of this mechanism of action. And if you can increase the potency by leaving the drug to incubate longer on infected cells, and the potency increases along with the efficacy, again, potentially a CYP51 inhibitor. And if you're able to go as far as the in vivo mouse model, then suppressive rather than curative activity. But this is really still under investigation at the moment. So the new screening cascade that we're working to at the moment is shown on this slide here. So we have an acceptance criteria for a new series, quite large, well, quite accepting in terms of in vitro IC50, below 5 micromolar, and host cell activity with an SI greater than 10, and typically good ADME characterization. We've got CYP3A4 in here just as an indication, maybe if we have a CYP51 inhibitor. I think that's just really a bit of a hangover from the efforts that we've run in previous years. But the new series profiling is something that we do to try and see what kind of, if we've got any new modes of action, and a really great assay that's been introduced into the field is a CYP51, cruzi CYP51 inhibition assay that was developed in the lab of Kevin Reed at Dundee University. And so you can actually measure directly using a fluorescence probe if you are inhibiting the enzyme. We do the trypanomastigote assay, and nice to have is a washout assay and a panel of strains but even now doing strain profiling in vitro and in vivo is really challenging and not on the main flow of the screening cascade. And fast killing, we're looking for fast killing actions, and so a time kill assay is also useful to do. And then on to your proof of principle experiments in vivo, which will be the in vivo assay in mice and tolerability. Okay, so how to choose the right start points? Well, if you think about where your compound's got to go to reach a parasite, there are many biological barriers and elimination mechanisms that protect intracellular pathogens from drugs. And so it's a really challenging design task. I think physicochemical and pharmacokinetic attributes are really important in this field, not just the potency. And I think that more lead-like type compounds, so low molecular weight, pretty polar molecules, possibly with weak basic centers, are the types of starting points that we need to be looking at in terms of being able to get drugs to site of action. And a really important aspect of compounds for T-cruzi inhibition is their high metabolic stability. So you really want your compound to be a brick and very resistant to oxidative metabolism, because each time we look at new series in this area, the ones with the high plasma concentrations over the time are really the compounds that show promising efficacy in vivo. And so we can maintain an open mind with regard to PK, but I've not come across a series where this observation hasn't held true so far. I also think there's opportunities to learn from R&D focused on intracellular bacteria. So two interesting papers here, one from a TB research effort looking at the complex disease pathology, and again postulating that small polar molecules are very good to aid diffusion into the cellular compartments. And there's obviously lots of work being done on the PK/PD of antibiotics for intracellular bacteria. So in closing, there's still lots to learn about the disease pathology of T-cruzi infection and Chagas disease itself. And the more we can learn, the better the assay design will become, and therefore the translation of promising molecules from bench side into the clinic. I think the requirements for new compounds to progress into the clinic are very stringent. So unlike visceral leishmaniasis, for example, we really have to be aiming for sterile cure, and we need to establish what assays will help us translate efficacy into the clinic. So we need to complete the cycle so we can validate our decision-making process with something that works. Having said that, there has been really rapid progress in the last decade, and this has been driven by a lot of pioneering researchers in the field, and really has been supported by the CYP51 compound story, which have been fantastic tools to drive this effort forwards, and they still continue to be investigated. But you know, is it time to switch to a new strategy? There's been a lot of phenotypic screening. It'd be great to see some target-based discovery efforts moving forwards. Perhaps some new mechanisms in the area as the basic research progresses, and maybe some new chemical modalities such as protein degraders and maybe next generation peptides to complement the work that's been done on small molecules. So millions of compounds have been screened against Trypanosoma cruzi, and the Chagas pipeline of potential new chemical entities is still very empty in contrast to leishmaniasis, for example. So we still have a way to progress. I think really I've highlighted what a significant challenge it is to develop new treatments for Chagas disease, but along with fellow researchers, collaborators and partners in the field, we need to keep trying to push back research boundaries, solve problems to get medications to the patients who need them the most. So with that, thank you very much to the great team of scientists I've worked with, and happy to take any questions.
Thanks for a great presentation, Martine. So really fascinating biology there. If anybody would like to ask a question, if you haven't got a microphone, please chat it. Please just type it into the chat box, and I'll put the questions to Martine. And if not, please, and if you have got a microphone and you just want to unmute yourself and ask a question, please feel free to do so. Just to get us going, I get the chairman's privilege of asking the first question. So, is there would it be possible to do a co-dosing approach and use the CYP51 inhibitor to reduce the parasite load and then knock it out with benzoyl, and therefore have a more tolerable treatment for Chagas?
Yeah, that's a very good observation. Yeah, that the co-dosing combination strategy has been investigated between benzoyl and posaconazole in assays, in vivo assays, and yes, I think there's a combination you can achieve there, and even if it's not with a CYP51 inhibitor, it may be that there are other drugs working by different mechanisms that a combination strategy would be successful for.
We have a question from Setcha. I'm going to unmute her. Well, I can try and unmute her. That's interesting. Setcha, can you unmute yourself? Yes. Oh dear, we can't hear you.
Yeah, I can hear you. Let me try.
Great, I can hear you too.
Can you hear me?
Yes.
Hello? Oh, can you hear me now? Is it better?
Yes,
okay.
You're clear.
I was talking about combinations. Yeah, I just wanted to ask if they tried dual or multi-target. Can you hear me?
Yes, you're a little bit difficult to hear, but
multi-targeted compound.
Do you want to type your question? And type your question. We can't hear you clearly.
Where one could be more than one in the past.
Yes.
Yeah. Okay, I will do so.
Satya, we can't hear you clearly enough. Can you just type the question? Oh, we have a question from Caroline Pete in Dundee, who says, we use EC90 of less than 1 micromolar against T. cruzi as progression criteria for our projects. Are we setting the bar too high at very early stages, e.g. series selection?
No, I don't think so. I mean, depends if you're left with any molecules at the end, I guess, after you've done your evaluation. But no, I think it's okay to be stringent at the beginning, and so that you're working on the molecules that deserve your attention. So no, I don't think that's too stringent. I think it's good practice to focus on what the best that you have to hand at the time, and then drive them to success or failure as quick as you can, and then you can pivot back to anything that was priority 2 that you might have dropped based on that criteria.
Question from Julian Pacheco. There are some evidence about the fact that amastigotes, I can't say that, can uptake cholesterol from the host. Do you think it might be related with the limited cure of posaconazole?
Good question. I'm not sure I can answer that one. I haven't come across that mastigoats can uptake cholesterol. I thought they were unable to use host cholesterol, hence being able to get some selectivity in vivo. But no, I can't comment on that, I'm afraid. I haven't read that research.
Julie, perhaps you'd like to send that to Martine.
Yeah, that would be great. Thank you. I'd be interested in reading that.
Yeah. So just out of interest, if you screened Kemble with your pharmacophore, how many compounds do you think you'd find?
Oh, just from experience, they're very, very easy to find, CYP51 inhibitors, and it's useful to have the fingerprint signature to be quite ruthless to remove them at the beginning. We're fortunate that we're able to run the compounds through the assay at DNDi just to check. Yeah. But yeah, if I see an aza heterocycle and something that looks a bit like a tripod, then I'm always suspicious.
Yeah. So do you think it's ever going to be possible to have to use a CYP51 inhibitor, or it's only ever going to suppress and not be something?
Look, I think you've got to keep an open mind, because we're still learning about disease pathology, and there is evidence that you can get fully efficacious CYP51 inhibitors. They've been measured in vitro. Mm-hmm. So I probably have moved on from CYP51 inhibitors, and it'd be good to diversify the modes of action. But I believe there are people who still work on them, and we know so little that it's hard you can't throw things away when treatments are so desperately needed. So if people can make them successful, that'd be great. But the evidence is at the moment for suppressive rather than curative activity if we're using the right assays to evaluate the compounds.
Any last questions? No? If not, I think we'll probably say a huge thank you to Martine. That was a tour de force in terms of explaining the CYP51 field. It will be really interesting to see if any new compounds start coming through, and it's clearly an area that's ripe for some new ideas. So thank you very much everyone for attending, and thank you Martine for presenting.
Thank you, Caroline.
I'll now close the meeting.
Lovely. Thank you.
Thank you.
Bye everyone.
Bye.